Assalamualaikum. My name is Muhammad Lutfi bin Ismail.
My name Siti Norazia Zuhra binti Mak Nasir.
I am Nur Aina Dalila binti Afandi.
In this video, we will talk about the genetic manipulation in biotechnology. So, let's get started. Picture this, you're making your lunch for class or work and you decide to pre-slice your apple because it's easier to eat and yeah, why not? Fast forward 5 hours, you're already exhausted from the day and all you want to do is eat your apple. But surprise, your apple slices are brown, a little shriveled and really just not that tasty looking. One of the most unappealing things about an apple can be the brown coloring caused by the bruising and slicing. Even though we know that the apple is fine to eat, the brown color make it kind of, well, gross looking. But what if this browning could be taken care of using science? It is all possible with genetic manipulation. So, what is genetic manipulation? Okay, I'm getting there. Genetic manipulation, also called genetic engineering or genetic modification, is a direct manipulation of an organism's genes by using biotechnology. So now we overcome the previous apple problem, but we must start from the beginning, which is from its plant. Genetic manipulation in plant involves adding a specific stretch of DNA into the plant's genome, giving it new or different characteristics, including changing the way the plant grows or making it resistant to a particular disease. The seed produced by this plant will inherit the new DNA, meaning by that it can be bred without changing it to the original plant characteristic. Back to the past, what is the first genetically engineered crop done by human using biotechnology? It is a tobacco crop plant reported in 1983 as the first genetically engineered plant and first developed by Michael Webster Bevan, Richard Bailey Flavel, and Mary Dell Chilton. They developed by creating a chimeric gene that joined an antibiotic-resistant gene to the T1 plasmid from Agrobacterium. They infected tobacco with Agrobacterium, transformed with an antibiotic-resistant gene, and through tissue culture technique were able to grow a new plant containing the resistant gene. The first field trial proved it's resistant to herbicide, making it easier to the farmer to remove the weed without kill their crop. In 1985, the genetically modified tobacco was introduced containing insecticidal protein from Bacillus thuringiensis that are insect resistant and has given a name BT tobacco. However, the crop was not commercially successful. But in 1995, the Environmental Protection Agency in the USA approved the commercial production and distribution of the BT crops.
So basically, there are two types of genetic manipulation, which are the genetically modified organism and transgenic organism. Genetically modified organism is the organisms that have had certain genes inserted or deleted, while the transgenic organisms is the organism that have been modified by bringing in the DNA of other species. And from that, we all know that all transgenic organisms are the genetically modified organism which had been modified at genomic level, while not all the genetically modified organisms are transgenic.
Okay, next is process of genetic engineering. Creating genetic engineering is a multi-step process. It is because gene must first chosen from insert into the organism according to the aim for the resultant organism. Next, genetic screening can be carried out to determine potential genes and further tests then used to identify the best candidates. Next is method of genetic engineering. There have 6 methods of genetic engineering that we will go through one by one. Okay, the first is isolate and clone of gene interest. As we learned before, DNA extraction is the first step in the genetic engineering process. A sample of an organism containing the gene of interest is taken through a series of steps to remove the DNA. Gene is separated by using restriction enzyme to cut the DNA into fragments or polymerase chain reaction (PCR) to amplify up the gene segment. Segment can be extracted through gel electrophoresis. Second is add DNA segment to initiate gene expressions. Once a gene has been cloned, gene combine with other genetic element before inserted into organism. In this stage, it has included a promoter and terminator which initiate and transcription. Third is add selectable markers. Gene transfer marker is needed to identify cell with new genes. Antibiotic-resistant genes are used as markers in order to replace an existing promoter with a new promoter. Okay, the fourth is transformation. Transformation is the transporting the recombinant DNA into the recipient cell or the host genome. There are various methods for recombinant DNA insertion is used such as microinjection, electroporation, biolistic, and vector-mediated. The main goal of each of these methods is to transport the new genes and deliver them into the nucleus of a cell wall without killing them. A biolistic method is one of the popular methods used in plant genetic engineering experiments. Biolistic transformation enables the direct introductions of DNA or RNA into the tDNA of the plasmid. The plasmid is reinserted into a bacterium. In this method, a foreign DNA or RNA are released from a gene gun by high-pressure helium gas and directly penetrate to the host cell wall. Next is select transform cell or tissue. The bacterium is used to insert the tDNA carrying the foreign gene into the chromosome of a plant cell. Then the plant cells are grown in culture. Okay, and the last one is regenerate whole plants. Transform plant cells are the regenerate into transgenic plants. A plant is generated from a cell clone. All of its cells carry the foreign gene and may express it as a new trait.
And now, let's move to the application of genetic manipulation. Crop protection is the main rationale behind the genetically modified crop, and the royalty of genetically modified crop is to produce one with disease resistance. Plants that are more resistant to disease spread by insects or viruses result in higher yields for farmers. Next, genetically modified could improve nutritional value in crop including edible oils, vitamin enrichment, and toxin reduction. For example, Camelina sativa are modified to produce plants that accumulate high levels of oil similar to fish oil. Genetically modified plants may someday be used to produce recombinant vaccines. Concept of oral vaccine expressed in plants for direct consumption by individual is being examined as a possible solution to spread of disease in undeveloped countries. Development of plant-denied vaccine in potatoes and lettuce for hepatitis B virus and Norwalk virus is underway to develop. Through genetic manipulation, the shelf life of a crop can be extended. These have been proved by many genetically modified products. Before, the Flavor Saver tomato was produced to have a longer shelf life. Besides, the United States Department of Agriculture has approved genetically modified potato that prevents bruising and the Arctic apple that prevents enzymatic browning after it has been sliced. Genetically modified crops are designed to tolerate to herbicides. As example, for soybean, Roundup tolerance conferred by expressions of a glyphosate-tolerant form of plant enzymes, the EPSPS, isolated from the soil bacterium Agrobacterium tumefaciens. Other than that, genetically modified plants have been used for bioremediation of contaminated soils containing mercury, selenium, and organic pollutants. For example, it is used in marine environment which are especially vulnerable since pollutions such as oil spills are not containable. As we all know, United States is the largest producer of genetically modified crops, and most of the genetically modified products in Malaysia are imported from the United States, and this includes the soy and the corn. Soybean with glyphosate herbicide tolerance comprises about 80% of global annual soybean production, and this ends up in our food, including the soy and corn oil, soy sauce, cornstarch, cornflakes, and many more. As for Malaysia, we are still at the early stage of biotechnology development and has not planted any genetically modified crop yet. However, we are experimenting to create genetically modified crops such as the bananas, papayas, and rice that are resist to virus infection, prolong shelf lives, and delay the ripening. We are also developing the genetically modified oil palm to increase its beneficial oil content as well as to increase the vitamin A and E in the oil palm production.
Next is limitations of genetic engineering. There are two limitations, which is lack of techniques for transferring multiple genes by transformations and complicating commercializations of a genetically engineered crop. It associated with the many of the tools of genetic engineering such as plant promoters and selectable markers. The cost of licensing these tools can be prohibitive, making genetic engineering currently feasible only for very high-value traits.
Genetic manipulation is a major scientific and technological revolution. However, this has created controversies and great debates. Some people say that the introduction of the genetically modified organisms to the biosphere bring the risk and could produce novel hazards. Most are afraid if the release of genetically modified crop will result plant becoming agricultural waste. People are concerned if the release of the genetically modified organism can bring any consequent harms to the environment, and that the foreign genes are spread and affect other crops. Besides that, having genetically modified organism as a food source also brings some social concern. In some cases, developing genetically modified crop by using gene from animal is an offensive act to vegetarian. Genetically modified crop could contribute to increase of food production and higher food availability, but may also impact on food quality and nutrient composition. People are also concerned about genetically modified food as if it might cause an allergic reaction or that are toxic.
We move to the National Biosafety Act 2007 and Genetic Modified Organism Product in Malaysia. Malaysia recognizes the twin aspect of modern biotechnology being the great potential offered by modern biotechnology and the need to protect human health and the environment from the possible adverse effects of biotechnology product. Biosafety Act is a procedure to provide harm to human health and environment with release of biotechnology product. Precautionary measures must be emphasized because the GMO has led to a serious debates as to whether these organisms may possibly create risk to the human health and the environment. In Malaysia, the Biosafety Act 2007 established a process to vet all applications for direct release of living modified organisms into the environment, including cultivation, content use, field trials, or consumption of GMO. Release activities also involve the research and development purposes in all field experiments such as placing in the market, offered as a gift, price, or free item, disposal and remediation purpose. The application for approval is vetted by Genetic Modification Advisory Committee, makes it recommendation to the National Biosafety Board for a decision. Any person who is found guilty shall be liable for a fine not exceeding 250,000 Malaysian ringgit or imprisonment not exceeding 5 years or both for an individual. As for a body corporate, they are liable for a fine of not exceeding 500,000 Malaysian ringgit. All GMO's product derived thereof are required to be identified and labeled to ensure products that have undergone the prescribed and to inform consumers about the contents of the product. Without this labeling, consumers do not know whether the food that they are consumed is genetically engineered or not. Finally, it may be concluded genetic manipulation is a scientific breakthrough that has led to developments in biotechnology. However, ethical issues arise as people try to determine if genetic manipulation crops are good or bad for humans and environment. Everything has the potential to be dangerous or harmful, including genetic engineering, although it could be benefit people in many ways. Therefore, the Biosafety Act is introduced by the government to control the activity safely. Thank you.